Yeah, sent you.
You have emailed me, is it?
Yeah, from my email to you.
Okay. Okay.
Yeah. Yeah, from my email.
ठीक है वो ना
അല്ല ഇനീഷ്യൽ വേണ്ട ഹിറ്റ് ആകുമ്പോ ഇതിന് ബയാസ്ഡ് ആയിട്ടുള്ള ഒരാളാണെങ്കിൽ കൂടുതൽ പ്രഷറാ വരിക. പക്ഷെ ഒന്നിനൊക്കെ പൊളിയാ.
ഇന്നലെ ആണെന്ന് എനിക്ക് അറിയില്ലായിരുന്നു. ഫ്രൈഡേ അല്ലായിരുന്നു.
Hello everyone, it's 10 o'clock now, but we're going to wait for a few minutes because a lot of people are still joining. So maybe by 10:05, 10:05 we will start. Thank you. Hello everyone. It's 10:05 now. We're going to start. Uh, my name is Vishwesha. I'm a associate professor at Center for Ecological Sciences, Indian Institute of Science. First of all, on behalf of everyone in our department and the organizing team, which has faculty as well as students, I would like to invite you all for this workshop, and we are also excited about you joining us today. So in the entire session, please keep your videos and audio muted. Of course, we expect that the workshop will be quite interactive, and then when you need to speak, when you want to ask questions, feel free to unmute yourself and speak. But in general, please keep both your video and audio on mute. And I want to, before I go further, I want to mention that the entire session is being recorded. It is also being live streamed on YouTube. So therefore, this is for your information if you have any concerns. Okay, so and also we will be recording all the sessions, and all the recorded sessions will be available for you to watch even after the event is over. So with that note, I will begin my introductory session now. So about the workshop first, why are we doing this workshop? We are our main goal is to reach out to diverse student pools and to create interest in ecology, evolution, behavior, and conservation research. And as you probably know, there are not too many undergrad programs that cover these really fascinating facets of biology, although they form the sort of bedrock of biological sciences. And in doing so, we want to introduce the basic research themes that are sort of current and important in these fields of ecology, evolution, behavior, and conservation. And not only do we want to introduce students to research themes, we also want to introduce various professional skills of being a researcher or being a scientist, because a lot of people are not really exposed to these. We all learn this on the fly over time, but these are really important skills too. So we will be covering workshops on these 4 topics, as well as many professional skills that will be very useful as a student, as a researcher, and a scientist. So the way we have structured the workshop is as follows. As you have seen from the schedule, this is a 4-day workshop starting from today and tomorrow. That's the first week of workshop, 19th and 20th of February 2021. And next Friday, Saturday are the other 2 days of the workshop. And on these 4 days, we run the workshops from 10 a.m. to 5 p.m. And when I say workshop, this is not really a lecture mode. We would like this to be as interactive as possible. And all the people who are going to conduct the workshop are trying their best to make the entire process as interactive as possible. And for that, we really like good participation from all of you. Okay, so now let me introduce briefly the Center for Ecological Sciences and who are all the people we will be interacting with over the next couple of weeks. And in Center for Ecological Sciences, we do research in the frontier areas of ecology, conservation, behavior, and evolution. And our laboratory is not restricted to small space in our department in our campus, but the entire Indian landscape forms our laboratory. And we have field station all the way from high altitude Himalayan areas to Western Ghats in the south, and in deserts as well as in Andaman and Nicobar. So we have a really wide range of field sites covering the diversity of Indian landscape. And we have 11 faculty members in our department and 2 honorary professors. Typically, at any point of time, we have about 50 PhD students every year 5 to 10 students graduate with PhD, doing really state-of-the-art research work. And in addition to PhD students, we have many BSc and master's thesis students. These may be from IASC, they're also from many universities outside. And in addition, we have many interns who typically come over in summer for 1 to 3 months. These are again typically BSc and MSc students. We will have a session conducted by students who will explain many aspects of our academic curriculum. So now let us see how the workshop is structured over the 2 weeks. The week 1 of workshop starting today each day has 2 sessions. And today we are going to begin with a workshop on evolution by Karthik Shankar, who is a professor at the Center for Ecological Sciences. I will not go into details of his research interests. He works on variety of taxa. Okay, and in the second half of today, I will be conducting a workshop on ecology, and my areas of interest are ecology and evolution, studying from a mathematical perspective. And then tomorrow morning, we have a workshop on behavior by Professor Maria Thakur. She studies, predator-prey interactions, animal movement ecology, and behavioral ecology broadly. And then after these 3 academic workshops in this week, the 4th workshop of this week, which is tomorrow afternoon, will be conducted by students. What does it mean to be a PhD student? How do we even begin applying and preparing for PhD programs? So the first workshop on how to apply for PhD programs will be done by CES students Nitin Saxena and Mihir Joshi. The second workshop tomorrow afternoon will on life of a PhD student. What does it mean to be a PhD student? How is it different from, let's say, being a BSc or MSc student? This will be done by a bunch of students, Pranoy, Avik, Kanika, Kajal, and others. And they will explain all these facets of being a student and also take any questions from you have. That's the plan for this week. However, and before we start the next week session, which is on next Friday, we will send you a bunch of videos from each research lab from CES. We will have a short video of 10 to 15 minutes duration, where faculty describe their research work in a short summary covering the basics and interesting aspects. And so we hope that by going through all these videos, you will want to interact with some of the faculty members and students. So what we will do over the next week is that we will also send you a form where we will ask you, would you like to meet any of the CES students and faculty? And please go through these videos that we will be sending to you by tomorrow evening, and then choose who do you want to meet. Okay, so we'll be sending more information on these videos and the form and the interaction section once the 2 days are over. So keep a watch on your emails, and then begins this week 2 of the workshop, which is on next Friday 26th and Saturday 27th. And next Friday, we begin with a workshop on quantitative thinking in ecology by Professor Kavita Ishwaran. Quantitative thinking and numbers and mathematics is not something we associate with biology, but as Professor Kavita Ishwaran will explain, this is really, really important. And then in the afternoon of Friday next week, we have 2 workshops. So one workshop is on communication skills that will be done by Dr. Harish Sridhar, who was a former student of CES and is currently at NCBS in Bangalore. And then in the afternoon, again the same day, we have a workshop on careers in ecology. So a lot of the times, students take various programs and join various programs without having a clarity on what are the future career options. We want to make sure that students in this group that we have invited are exposed to these early on. So therefore, you can learn what do students do after PhD. So we hope that this will be exciting to you. And then on the morning of 27th, the last day, Professor Umesh Srinivasan will talk about conservation research. He works on various aspects of forests and global climate change. This will be next Friday morning at 10 a.m. Next Saturday morning, sorry. And then finally, the last session of the workshop will be faculty-student interaction session. As I mentioned, we would have sent you the forms we would be you pick which faculty do you want to meet based on your preferences and the availability. We will put you on interaction sessions with faculty members, and then finally we have a concluding session on the 27th February. So that's the plan for the 4-day, 2-week long workshop. And in addition to the faculty members I have introduced, we have many other faculty members at CES. Karthik Sunagar works on evolution of animal venom, ecology, evolution and of animal venoms. Again, we hope you will be able to see his video. Dr. Sumanta Bagchi works on ecosystem, structure, function, and services. And then Professor Rohini Balakrishnan works on acoustic communication in insects and bats. And Professor Rene Borges works on plant-animal interactions, from fig wasps or associations to termites and many other systems. And Professor Praveen Karant works on biogeography and evolution of various taxa on the Indian subcontinent. He's also the chair of our department. And finally, we have 2 honorary faculty, Professor Raghavendra Gadakar and Professor Sukumar, who have done phenomenal work in their areas of research and are internationally very, very well known. And then we have a new faculty joining very soon, Professor Saskia Newhouse. And I'm not very sure if we will have any videos from these faculties. Okay, so in addition to various faculty members and research, we have a very good curation of specimens in our department. We have herbarium and museum, and then we have many facilities from computational to molecular and environmental chemistry laboratories. In addition, CES also coordinates long-term ecological observatory program. So that's the brief introduction to our department and the workshop today. These are all happy and fun students of CES. So with that note, I'm going to now stop and get back to the program for the day. And as I reminded you, we will share videos of as many faculty members as possible by tomorrow evening. And so let me now come to today's program, where we begin with a workshop on evolution by Karthik Shankar. So I will stop here. If you have any queries, questions, there are 2 options for you to coordinate and contact us. One is chat box. So I think most of you should be able to see that there is a. So what I'll do is I'll just show you by sharing the full screen how you can use the chat box. Karthik, hold on for a second. I'll just. Okay, are you able to see now? Are you able to see my screen?
Yes,
Are you're seeing my program again? No, I didn't want that. Okay, I'm just going to share the. Okay, I don't want this. I wanted to share the Zoom. I wanted to share the Zoom screen. I can't do that. Okay, fine. I'm going to skip this. Okay, so in the center of the bottom panel on your Zoom, you should see a chat box. So you should be able to write any questions you have. And in addition, there is a feature called Raise Your Hand. Okay, do all of you know where it is? So again, on your panel, there is something called Reactions. And there's something called Raise Your Hand. If you raise your hand, it shows up and we can then ask you to speak up. So if you like to speak up, please raise your hand. If you like to just send a question by chat, just message in chat. One of us will read and answer your question. Okay. So over to you, Professor Karthik Shankar.
Thank you very much, Vishu. That's a lovely introduction. Lovely to see so many people here. We're sort of delighted to conduct this workshop. And I would also sort of request the volunteers for this session to keep an eye on the chat box and on raised hands, because I might not notice while I'm talking. Yeah, so let's get started. As Professor Guttal said, we'd really like this to be interactive, so feel free to raise your hands, ask questions, and I've prepared a certain amount of material that I'd like to share today. but we don't have to go through all of it. We'll take it at the at the pace that that we go at. And as you can imagine, we're each of these workshops that we're conducting, ecology, evolution, behavior, each of these is an entire course by itself. So we're sort of trying to compress some important ideas within a couple of hours. Let's see how much of that we can do. All right, let me just share my screen with you. Yeah, the host's gonna have to allow me to share my screen.
Arun, like, do you need to make Karthik co-host?
So while we're waiting for that, I'm going to start by asking you a question and giving you 1 minute or less, let's say 30 seconds, to write down in 10 words what evolution is. I'm not going to ask you to share that because we'll be here all day, but for yourself, I'd like you to just write down in 10, 12, 15 words, what is the answer to the question, what is evolution? Don't use the chat box. I want everybody to do this for themselves without access to what other people might be thinking about it. So just write down in about 10 to 15 words for yourself what you think evolution is. Yeah.
Karthik, you must be able to share now.
Yeah, sure. I can. And you're going to do an interesting exercise for yourself. This is entirely for yourself. Don't use the chat box. Write it for yourself. Now at the beginning, and then at the end of the workshop, see if you want to modify that in some way. Okay. Yeah, and don't send me direct messages either, because while I'm giving the talk, I'm not going to be able to read them. So this is not an evaluation. This is just for yourself to know what your definition was at the beginning and what your definition was at the end. Let me share my screen now. Okay, can someone confirm that you can see my screen?
Yes.
Yes, sir.
Yes, Karthik, we can see your screen.
Excellent. All right. Okay. So we're going to be broadly talking about both pattern and process in evolution. Yeah. And we shall start by asking why we're interested in this. So if we look at global patterns of diversity, for example, you will find you can draw maps of global amphibian richness. The red is high diversity, and as you can see, with amphibians, which are frogs and salamanders and caecilians, the highest density, the highest diversity is in South America, tropical Africa, and tropical India and Asia. Now, you can similarly look at a map of global fish richness or global plant richness, and in each of these cases, you will find exactly the same patterns. Which leads us to ask this question: what causes diversity? What is the cause of these areas that have these incredibly high levels of diversity, which are known as biodiversity hotspots, relative to the rest of the world? Or more specifically, we can say, why do some areas within the tropics have greater diversity? Now, before we get into that, we can think about diversity at two levels. We can think about diversity in what are called clades. A clade is a group composed of an ancestor and all of its descendants. And we can ask that question: why is the diversity of some clades greater? Now you take two groups. One is the Tuatara to the left, which is evolved originally 200 million years ago. And over that period has only had 10 species, of which only one species is still alive today. And then you take a group that is roughly, in fact, even younger, just about 150 million years old, perhaps, of birds, and this has 8,000 species. Similarly, you have groups like snakes, which have 3,000 to 4,000 species, which are again just about 150 million years old. So why are some groups of organisms as old as the Tuatara and have so few species? And why do other species, other clades like birds and snakes and lizards have so many species? Similarly, we can ask about diversity in space, right? Now, in space, you have certain groups such as Hawaiian honeycreepers, African cichlids, Caribbean anoles, the lizards, plants such as bromeliads, other birds such as the wangers of Madagascar. And these are all groups of animals or plants that have very high diversity, but they're also concentrated in particular areas, right? So we can sort of think about diversity as a combination of being a pattern in a clade that is along a evolutionary lineage, or also the biogeographic processes such as dispersal, right? So what we see as these biodiversity hotspots or these what are called radiations is really a combination of diversity in time and space, diversity in time through evolution, and diversity in space through biogeographic processes. But there are a few other patterns that I want to bring to your attention before we talk about broader level patterns and processes. Let's, for example, take frogs, which we study a lot in the Western Ghats. And I want to introduce you to a few groups of frogs which we've studied over the last 10 or 15 years, which show some very interesting patterns. This is a group, for example, of bush frogs or the genus Raucestus, which is found in the Western Ghats. And what's really interesting about these bush frogs is that you have many, many species which all look dramatically different from each other. There are bush frogs that look similar to each other, but you take another group that we study, that several amphibian biologists in the country have studied, such as Nyctibatrachus or wrinkled frogs, and you find that typically these look very much like each other. In fact, you have many similar bodied species that you can't even tell apart. And so you have these patterns of two groups of frogs that diversified in the Western Ghats. One has about 60 species, the other has 40 to 50 species, so lots of species. But in one case, you get lots of morphological divergence, and in the other case, you find very little morphological divergence. Right? So they may have been evolving for the same amount of time, they've evolved into the same number of species, but sometimes you get a lot of divergence and sometimes you get very little. There's another very interesting pattern. Now, if you contrast those two groups, Nyctibatrachus has a closely related other genus called Astrobatrachus, which was just discovered and described just 2 years ago. Now, even though these are so closely related, Astrobatrachus just has one species. So similar to my question about tuataras and birds, why has one group been evolving in the Western Ghats for the last 30 million years and may have 40 to 50 species, and another group, which is perhaps even older, just has one species in it? An even more striking example is this purple frog, which was discovered about 15 years ago, has its closest relatives in the Seychelles, probably diverged about somewhere between 60 and 100 million years ago, again represented by just two species in the Western Ghats. So you get all these really fascinating patterns, and that then makes us ask the question, why do these patterns come about? How do they come about the way they do? And how do we try to understand it, right? So when I say how it all began, you probably think I'm going to talk about how life began on Earth, but actually we're here as people interested in evolution as either evolutionary biologists or aspiring evolutionary biologists or students of evolutionary biology. And we have to acknowledge that our work today stands on the shoulders of giants, starting with some naturalists nearly 400 years ago. And the reason I start here is because the study of evolution in a sense, or the study of form and function or adaptation, really started with those same questions that I gave, those same patterns that I posed to you at the outset. Now, I said there are these global patterns of frog richness or bird richness or plant richness. Why do these come about? Why are there so many species? And why are there so many species in certain areas? Now, the same questions affect, influenced John Ray, who was a lecturer in humanities and Greek and mathematics at Cambridge, but he got really interested in plants. Now, at that time, plants were only classified by their relationship to man: edible plants, poisonous plants, annoying plants, and so on. And he started slowly cataloging them much more systematically using morphological features. And when he developed his catalog of Cambridge plants, the number grew from hundreds to thousands. And with his friend, Francis Willoughby, Willoughby was very important, though he was a Ray student. Willoughby had money, and as you will all discover during the course of this workshop, funding is very important. Ray had Willoughby's money to spend, and so they also traveled around England studying birds. And they then wrote a book called Ornithology, where they classified birds based on form and structure. It was the first time in the Western scientific world that in natural history, people had started using a systematic approach involving things like form and structure for classifying plants and animals. Now, this led to, well, before I go further, well, for those of, for all the women in the audience today, you can be inspired by the naturalist Maria Sibylla Merian, who actually, again, in the late 17th century, pioneered a lot of natural history. And she traveled around the world, on her own, in fact, with her daughter, and was the first person to describe the metamorphosis of the butterfly. And people like Ray and Merian and Willoughby made these early, very important contributions to natural history, which was then consolidated by someone whose name I'm sure you've all heard, Carl Linnaeus, who's mainly known to us as the father of modern taxonomy. But Linnaeus also made a couple of very, very important contributions. People at that time believed that new species can come about in different ways by different animals breeding with each other. Some people believed that mice came out of the earth, and so on. Linnaeus posited that every species was bound together by reproduction, that no new species were produced at that time, that species were real units, and then he produced a system of classification for them. And he also emphasized the importance of ecological relationships. So we start with the description of diversity in the world around you. Then you learn to classify that diversity. Then you recognize those importance of ecological relationships. But what's happening in this 100-year period is this very important question is being asked, or important questions. Why are there so many different forms? Why do some birds have curved beaks? Why do some birds have straight beaks? Why do birds have different kinds of claws? Why are there so many kinds of leaves, right? And also, by then, people had started to discover fossils and were realizing that species were beginning to go extinct. And so this whole question of the formation of new species, of evolution, had started to come about by the late 18th century. I'm going to skip a lot of the history and move straight to the early to mid 19th century, where, as you may have read, Charles Darwin undertook this voyage on the Beagle, made extensive documentation of all kinds of plants and animals and rocks, documented the variation in the finches of the Galapagos, which are known as Darwin's finches today. And at the same time, Alfred Russel Wallace was undertaking travels to the Malay archipelago, and naturalists of the time, such as Charles Lyell and others, were debating this question. They knew that new species came into existence. They didn't know how. Darwin combined the idea of natural checks with his learning about population growth from Thomas Malthus's essay, and saw the struggle for existence as a powerful selective force, which he named as natural selection. And in 1858, Wallace came up with the same idea, which Darwin described. He said, Wallace had read my sketch. He could not have made a better abstract. The scientists, the naturalists of the time, arranged for them to read their paper together at the Linnean Society. And that sort of was the beginning of our understanding of evolution by natural selection. Genetics was, of course, discovered many, many, many years later, which allowed us to synthesize our knowledge of the mode of inheritance with the ideas of natural selection that Darwin and Wallace posed, right? So in a sense, the questions that we ask today, we look around at this diversity and we see, why do we see what we see outside our window? Why do we see what we see in this forest we go to? Come from the same history of human inquiry among naturalists over that 200-year period, starting with recognizing diversity, asking about their ecological relationships, asking how selection and adaptation occurs, leading up to the theories of Darwin and Wallace. Okay, so broadly, before I sort of like today's talk is, as I said, we're going to focus on pattern and process. And we can think of evolutionary biology as comprising two major endeavors. One is inferring the history of evolution, and the other is looking at microevolutionary processes, which are the sort of the changes in genotypes, the changes in alleles, and how these changes in alleles lead to modification in populations, and eventually to the formation of new species and to diversification at a larger scale. So let's start that out by looking by the first broader process that we talked about, in inferring evolutionary history or evolutionary relationships, which is really the study of how organisms are related to each other, which also leads to to systematics or the classification of organisms. Now, let me ask you a question. Let me pull up the, yeah. So, are you all familiar with the classification of vertebrates? You will the major classes being fish, amphibians, reptiles, aves or birds, and mammals, right? So can you draw a tree again for yourself? I'll give you a minute to do that, which shows the relationships between fish, amphibians, reptiles, birds, and mammals. Let's take 10 seconds to do that.
They can type in chat box, right?
Yes, you can type it in the chat box if you like. I'm going to ask you in a second if any of you would like to put your hand up and describe that to me. That would be fine. So let me propose that as a simpler question. Who are the closest relatives of mammals? Question number 2 is, who are the closest relatives of birds? You can answer that in the chat box, or you can put your hand up. I see a raised, is there a raised hand there? Do you want to be more specific? Who are the most closest amongst reptiles? Who are the, well, so everybody knows that birds and reptiles are very closely related. Great. Now, who would you like to tell me within reptiles are birds equally closely related to everybody? Or are they particularly closely related to some reptiles more than others? Rintu says crocodiles and birds. Great. So we seem to be getting, some kind of a little bit of a consensus over here. A lot of people agree that reptiles are closely related to mammals. Reptiles are also closely related to birds. Great. Now, you also said that birds and crocodiles are very closely related. So, whoops. Let's go back. Yeah. You guys are absolutely right. Now, 20 years ago, there was debate about the exact nature of these relationships between turtles and crocodiles and birds and so on. But predominantly, the work, much of today's the work so far indicates that birds and crocodiles are really closely related. Now, this sort of begs the question, what, if anything, is a reptile, right? Now, either those classifying vertebrates should have classified birds as, as a subgroup of reptiles, or all of those groups should have been classified separately, and that birds and crocodilians as, as a group together. So sometimes this is a particularly good example of the classification that we carried out on the basis of morphology, not really reflecting the evolutionary relationships between these groups. In fact, it's fairly well known today that the early ancestors of mammals and reptiles diverged into mammal and reptilian groups. Some of the groups that evolved into dinosaurs, in fact, gave rise to the birds as we see them today. So if someone says there are no extinct dinosaurs, there are. They're called birds. And often the grouping that we make in terms of amphibians and reptiles, in fact, they call herpetofauna, and this is called herpetology, makes no sense at all from an evolutionary perspective. Another example is of vultures, and typically the classification suggested that all of the New World and Old World vultures were related to each other, which were then related to other birds of prey and storks and so on. But what evolutionary relationships based on molecular genetics shows that, in fact, that the New World vultures are more closely related to storks, while the Old World vultures are related to birds of prey. So much of the history of much of our work in understanding evolutionary relationships, which of course for 200 years was based on morphology, and in the last 2 or 3 decades on genetic data, is on better understanding these evolutionary relationships between organisms. Now, in that, there are 2 things that have come out over here in these few examples that we looked at, and one is the issue of convergence, which is when unrelated organisms evolve structures, traits, or features that have the same functions. And as you know, birds, or well, in this case, an ability to swim from fish to dolphins to birds such as puffins or ichthyosaurs. or flight, the wings of numerous different kinds of animals, from birds to bats to gliding reptiles and so on. Many extinct flying reptiles as well. But in terms of understanding the processes and history of evolution, in fact, I would say our starting point would be to understand divergence, which is characters which are shared and then have diverged either in adaptation to the environment or due to other factors, which we'll come to a bit later. So let's keep these ideas of convergence and divergence in mind, and go to one more example that I want to share with you. Now, going from 1 to 6, 1 is a shrew, 2, let me think. 2 is a kangaroo, 3 is an elephant shrew, 4 is a hyrax, 5 is a manatee, manatees and dugongs, or sea cows, and 6 is an elephant. Right? I want you to put these into groups and tell me which the closest relatives to each other are. Right? You can create more than one group. So if there's any animal that's really closely related to any other animal in this relative to the others, I want you to put those into a group. Like, for example, if I were, I might look at this and say, I think elephants and kangaroos are really closely related to each other relative to everybody else. Now, I can also tell you that we all probably know that's not correct, but that's the kind of grouping that I want you to make. So going from 1 to 6 again, 1 is a shrew, 2 is a kangaroo, 3 is an elephant shrew, 4 is a rock hyrax, 5 is a dugong, a manatee, and 6 is an elephant. Excellent. Shrews and elephant shrews. You're using the morphology of language there. I would have guessed the same thing. Shrews and elephant shrews should be closely related. Manatee and elephant, good guess. Shrews and elephant shrews, why not? Shrews, elephant shrews, hyrax, kangaroo, elephant, and manatee. That's an excellent answer. That's what people thought for many, 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 many years. Well, not exactly that, but shrew and kangaroo, because they rhyme. I like that as well. So, you know what we're doing right now? We're building phylogenetic trees. We're building evolutionary trees. We're building evolutionary trees on the basis of different things. We're using a combination of some of you are using morphology. Some of you are using well, maybe there's a little hint in the name. The elephant and the manatee have a similar appearance. The shrew and the elephant shrew have a similar appearance. So we're building these trees very quickly and rapidly on the basis of something we know about how they look, which you're seeing in these pictures, or maybe something you've read about how they behave, or something you've read about, about the history of mammalian evolution, and so on. Right. So what we're actually doing is building evolutionary trees, and that's great. Now, I give this example because I want to sort of like precede that by saying that some of the like the evolutionary groupings or the groupings of classification that Aristotle did 2,000 years ago continue to be correct till date. What John Ray and Willoughby and Linnaeus classified, Linnaeus was the first to classify humans as Homo in a separate genus by ourselves. and what Darwin and Wallace and many others did, a large part of classification on the basis of morphology has in fact turned out to be correct. But mammals are really interesting. If you were classifying on the basis of the name giving you a hint, what you should have done is classified elephant shrews and elephants together. It came as quite a shock to evolutionary biologists when 20 years ago, when they carried out a molecular phylogeny of mammals and found that elephant shrews belong to a group that they classified as Afrotheria of African mammals, which included, golden moles, tenrecs, elephant shrews, which had all previously been classified in this group with shrews and their relatives. It also included aardvarks. And then, of course, it included manatees and elephants, as many of you pointed out, but also rock hyraxes, which actually hyraxes had long been known to be closely related to elephants, even before the molecular phylogenies were carried out. So as you can see, when we build these evolutionary relationships, we get some really fascinating insights into the history of how life evolved on Earth, the relationships between different groups of animals and in their classification. So this is one of the first things that we infer about, that we use evolutionary biology for, to infer broad-scale evolutionary history, to understand the relationship between different groups of animals, and sometimes find these really, I think, ridiculous things where I can tell you that in the 200 years that people had classified shrews and mole shrews and elephant shrews in one group as insectivora, which was still very much how it was understood when I was doing my bachelor's or master's or even my PhD. Insectivora included all those, and it was just called elephant shrew because of its long snout. And this group in the US carried this large-scale mammal phylogeny based on genetics, and I think the paper had just come out in 2000 and perhaps in Nature, and we were all so shocked when we saw that actually an elephant shrew was closely related to an elephant. So that's kind of a cute story, but there are many similar stories, many surprises for us in building these large evolutionary relationships. Okay, so before I move to the next sort of section, do you have any questions? This is the slide. I'm hoping you can see it, right? Like the tree at the bottom. Marsupials are the most distantly related. They're a separate group, and all of these others that are clubbed together over here in the middle of the screen are your. Where's the pointer? Pointer options.
Karthik, you have unshared the slide, so I don't think right now people can see.
Okay. Hang on. Let me share it again. That is not the one that I want to show you. Okay. Can you see now?
Yes, sir.
Yeah. So this, I'll just leave that with you for a second while I wait to see if there are any questions. Marsupials or kangaroos are the most different lineage from all other placental mammals. So mammals consist of three large groups: the egg-laying mammals, such as the platypus and the echidna, and the marsupials, which are found only in Australia and South America, and placental mammals, which are found across the world. I don't actually show you all of the other mammals. As you can see over here, the The group that I'm sort of focusing on is Afrotheria, which has, as I said, all of these, including the elephant shrews and other moles and tenrecs, and another group comprising manatees, hyraxes, and elephants. The other groups of mammals include primates, rodents, rabbits and their relatives, shrews and their relatives, and all other mammals, which is all of your carnivores and ungulates and bats and all of them. All right. Are there questions in the chat group, Gokul or Kanika, who was volunteering?
It is actually one
No
question
questions.
here.
Huh? There are questions? Let me stop sharing then. Sorry?
So question by Deepali.
Yeah. Can you just read out the
Question by Deepali is that how do you come to know the evolutionary relationships between organisms?
Right. Let me just go back to my screen and I'll have someone read out the questions for me. That would. Okay, so historically, so as I, when I was talking about convergence and divergence, what we're interested is in factors or features that reflect a trait that an organism had that it shared with another organism because of their shared evolutionary history, right? Now, you can think about that in terms of, let's say let's take an example of humans, right? Now, did I inherit my the color of my eye from my parent, and do my sibling and I have that same trait because we both inherited it from that parent, because the parent had that gene for, let's say, the blue eye or like a brown eye or a green eye, and both my sibling and I inherited that gene, and therefore we have that trait, right? And this is a morphological trait. Now, one can also measure that at the genetic level itself to say, is this a copy of a gene that I inherited from my parent? Now, if we look at either morphology or genetics on an evolutionary scale, that's the kind of question that we're asking in terms of evolutionary relationships. So, for example, do two species share a particular morphological trait because they inherited it from a common ancestor? That's what we're looking for in building the evolutionary tree, right? Now, because in morphology you can get convergence, which is that morphological traits can evolve in adaptation to something, they may not always reflect that evolutionary history, which is why sometimes when we build these trees based on genetic data, you get results that are counterintuitive, like the example that I just showed you. But with genetics as well, what we're looking at is mutations in the DNA, which we look at DNA sequences and we look at mutations in that, which reflect a shared evolutionary history. So you have a sequence of DNA from a particular part of the genome. It could be from the nuclear DNA, or it could be from mitochondrial, and we're comparing two sequences. So as an example, we might be comparing two sequences of mitochondrial DNA from, say, a cytochrome B gene, for example. I mean, and there are many such, and you're looking at those and looking at the level of similarity. And you would expect changes to accumulate over time, and therefore those that are more closely related would have more similar sequences, and those that are more distantly related would have more dissimilar sequences. And we use that information. There are different kinds of approaches to build. Yeah.
Hi, Karthik. We have another question from Dheeraj.
Yeah.
So his question is, are elephant shrews more closely related to African elephants than Asian elephants?
That really isn't, is a good question, simply because we're like, right now anything is possible, right? So no, so the group, the all of elephants evolved together, and then elephants diverged into African and Asian elephants, and Asian elephants elephants colonized Asia subsequently. So Asian elephants are a sister group of African elephants. They're very closely related to African elephants. As you can see over here, you can see my, oops. Yeah, am I still sharing? Yeah, I'm sharing. All right. So if you can see my pointer over here, if I bifurcated this into two more, one of them would be the African elephant and one of them would be the Asian elephant. Actually, there would be many extinct elephants as well as in that group. Okay. I'll sort of like during the break, I'll look at the rest of the questions as well and answer them. Okay. So this is one part of it, right? Who's related to whom, who's not related to whom, which two of those people, those are twins who look very different, or those are siblings that look the same, all that sort of thing we're looking at in an evolutionary sense. But that's sort of like evolutionary relationships over time. Now, at the same time that all of this evolution was taking place on Earth over the last 500 million years, 600 million years, the Earth was changing, right? The Earth was not constant. Now, if you look at over that very large sort of period of 300 plus million years, the continents have actually moved in relation to each other. Now, the person who came up with this was Alfred Wegener, who came up with the theory of plate tectonics, which said essentially that this is a complex interaction of the Earth's crust, mantle, and core, and it consists of plates that are about 100 kilometers thick, which sit on top of this rigid layer of crust, which adheres to this mantle, and then they move around because of a set of different forces. And let's not worry about these forces now, but essentially what this is like is a bit like suitcases on a conveyor belt, right? And they're all moving along and pushing and pulling at each other. And what has happened, therefore, over hundreds of millions of years, is that the continents have moved in relation to each other. The ones that have been, important in much of the history of life on Earth in the last 300 plus million years have been Pangaea, Gondwana, and Laurasia. And Pangaea was a supercontinent that existed briefly about between 300 and 200 million years ago. And then it split into these large continents of Gondwana and Laurasia. Now, how do we know this, right? One of the evidence for this theory, in fact, came from the distribution of plants and animals. And one of the most marked of this is an extinct group called the Glossopteridales, or the Glossopterus flora, which you find across parts of South America, Southern Africa, India, Australia, and so on. And why you find them in these particular areas is because these were all in contact with each other at one time. Now, the Glossopterus fauna largely went extinct during the Permian extinction, which is at the end of the Paleozoic era. There was this huge mass extinction where 80-90% of life went extinct, and most of the Glossopterus went extinct then. But there are other fauna as well, such as Carabidae or ground beetles, Proteaceae, which are flowering plants, Pipidae, which are these really strange frogs. And as you can see in each of these, the distributions of all of these, they look very weird, right? If the continents had always been the way they had been today, we'd be like, why are they found in these particular locations? But if you actually think of these as having all been together at one point in time, as Gondwana, for example, which then subsequently broke up, then it makes sense, right? At that point of time, 150 to 200 million years ago, they were all in land masses that were connected to each other, and then they separated over, between 100 and 200 million years ago, and then India started moving away and eventually floated across the Indian Ocean and joined with Asia. I couldn't help throwing that in. We don't often think about dinosaurs, but there's been a lot of wonderful paleontological studies in the last 40, 50 years, and a number of dinosaurs, such as Parapasaurus, is the largest known sauropod known from India. Was believed to go to 15 to 20 meters in length and weigh several tons. But there are many other dinosaurs from India, such as Stringosaurus and Genosaurus, and all of which kind of like lived in this floating continent that was moving, well, even before it left Gondwana, but then as it floated across, many dinosaurs have been discovered from different sort of paleontological sites across India. So if you think about how that might have, how to visualize that, it might have looked something like this. And I'm going to play that for you one more time. That started out as a supercontinent Pangaea, which existed till about 200 million years ago, and then started breaking up like so. And then by about 100 million years, Gondwana's more or less broken up, and India starts moving towards Asia, makes contact sometime between 50 and 30 million years, 55 and 35 million years ago. And by about 35 million years ago has fully established contact with Asia, and resulting in the Himalayas. It's nothing that I would want you to sort of like remember, but just to sort of place that in that context, from the Paleozoic period extending from about 250 to 600 million years ago, was at the beginning of the Paleozoic in the Cambrian was the big diversification, followed by the earliest fish, the invasion of land by vertebrates, the evolution of the first reptiles and mammals, the great extinction at the end of the Permian that I mentioned, and then through the Mesozoic, the diversification of dinosaurs, which were the largest, some of the most diverse groups ever to have evolved and survived for 150 million years. Through the Jurassic and Cretaceous, the extinction of the dinosaurs, the domination of flowering plants, and so on. And then the last phase, which is the Cenozoic, which is when India and Africa both collide with Eurasia. There's another major extinction of marine invertebrates, but also the diversification of land mammals. So all of this happens in that, on the stage of all of those continents moving across. So there's the diversification of these different groups. Some go extinct, some diversify. All of this happens while these continents are moving across space over that period of time. So this is sort of like a broad, this is okay, life on Earth, as you might see in a serial. But how do we as evolutionary biologists try to understand and study that with respect to the, to the plants and animals that we are interested in? And one of the baskets of that I would put that in is what we call biogeography, which is or historical biogeography, which is understanding the history of the evolution of these plants and animals across this space, across this this globe, where all of these land masses were moving relative to each other, experiencing different environmental conditions, and so on. And I like to sort of frame that by saying that we can think of where species are found today as being influenced by three things. It's influenced by where it originated or where it evolved. or it evolved somewhere else and it got there by dispersing there. I'm going to talk about all these processes later. But once it got there, it also had to stay alive, right? In the sense that it had to survive the environmental conditions, perhaps competition and predation, and so on. Now, these processes, which let's focus on these, which are dispersal and diversification, can be thought of as the movement of organisms from one location to another, which can happen in a few different ways. It can also happen by the origin of one or many species from pre-existing ones through processes that are called adaptive and non-adaptive radiation, which are radiation essentially being the formation of many species. Adaptive is when there's a morphological trait that has evolved in response to something in the environment, as in the classic case with Darwin's finches. But in other cases, this radiation also happens due to geographical isolation and doesn't necessarily involve morphological divergence. And many research groups have spent the last several decades studying the biogeography of the Western Ghats. And I wanted to give you a few very specific examples from our own backyard. And we can start by asking these same broad level questions about how did species enter India? Did they come from Gondwana, or did they come from Asia after India had collided with Asia? So, for example, if we collected those taxa that we're studying, they could be lizards or frogs or birds or snakes or plants from these different locations, and conducted a molecular study, we could then build up a phylogenetic tree, which from my colleague in CS, Professor Karanth, and his former student Aniruddha Roy, in their study showed these really nice sort of depictions of what you would expect if you had Gondwanan origin of any taxa this could be, where you would expect that the species in India would be related to those in Madagascar, as you can see in the graphic above, that the closest relatives of the Indian and South Asian, in this case, skinks, I imagine, were the ones in Madagascar, right? So the ones in Madagascar are closely related to the entire group because they've come from Madagascar to India, and then when India collided with Asia, those have gone into Asia. Now, you have an alternate scenario where the species from Southeast Asia came into India, where the species in India are most closely related to those in Southeast Asia, and those then as a group are related to those in Africa, which are all most closely related to each other. So you can have different scenarios of divergence, which different kinds of evolutionary trees or phylogenetic trees will tell you who's most closely related to whom and how this dispersal or diversification may have taken place. We can ask that question more specifically about the Western Ghats. We can say, how did they arrive in the Western Ghats? I know they came from Southeast Asia, but maybe they came through the Satpura Hills. It's called the Satpura hypothesis, or maybe they came through the Eastern Ghats. Maybe they came through the Himalayas and the Aravalli. So there are many theories about how Southeast Asian fauna may have reached the Western Ghats. And these are the kinds of questions that we answer using molecular data and field studies. But I just wanted to emphasize before showing you the results of those studies that when we carry out studies like these, either at the scale of the Western Ghats or peninsular India or all of India, they involve extensive field collections. So, for example, in this field study of the Western Ghats that we carried out over a 10-year period, all of the mountain complexes, the 14 different mountain complexes, all of these were visited, and collections were made of frogs and snakes and lizards, and across all of the elevational gradients, across all of the habitat types, and so on. And of course, if you're doing this at even larger scales, like all of India, then you have to cover those even larger geographical regions. So some really cool results came out of our work as well as that of others. this group called bush frogs or Alcestis. There are now 60 odd species of these known. Some of these look very alike and some of them look very different, as I mentioned. But one of the cool things was that we found two distinct groups. One which we call the north clade and one which we call the south clade. The north clade are all species that are closely related to each other, more closely related to each other. The south clade are almost more closely related to each other. And these were not only were they divergent from each other, they'd separated out, and they were separated by this thing called the Palghat Gap, which is the deepest valley in the entire Western Ghats range. So this Palghat Gap has been dividing species for 15 to 20 million years. Another interesting thing was this northern clade had many open canopy or grassland adapted frogs, whereas the southern clade had more adapted to closed canopy, and there were more species, in fact, in the mid-story and the canopy of trees, which were not found in the northern clade at all. So you can see that the evolutionary history, that these are diverging from each other, and that the species in these different clades are also showing adaptations to different habitats that they were exposed to. You see a similar pattern of there being a strong north clade and a south clade in these Nyctobatrachus or night frogs that I showed you earlier. See a similar pattern in agamid lizards, except with agamid lizards, you also find that there are species in peninsular India and the Western Ghats that are also found across all of peninsular India, the Indian subcontinent, as well as those are closely related to those in Southeast Asia. So these are actually lizards that came in from Asia, and some of them diverged and diversified within the Western Ghats. And again, similar to the lizards, but a little in contrast to the frogs, with snakes such as vine snakes and pit vipers, where you have a group that's Southeast Asian in origin, which also has some species in India. And then there's another group or another clade shown here in blue, which are species that diversified entirely within peninsular India, and some of them entirely, in fact, within the Western Ghats. But not just frogs and snakes and lizards. Our colleague Vivi Robin showed similar patterns with birds. Surprisingly, you'd think that birds could fly everywhere and they'd fly across this gap quite easily, but laughing thrushes, which is the tree to the left, Montesincla, and the shortwings or robins, which are the group to the right, which they named as a new genus called Sholicola. These also showed these divergences. The populations above the northern Western Ghats were separated from the ones below the southern Western Ghats. And in fact, even below the southern Western Ghats, there was a difference due to the barrier of the Shenkota Gap. Like birds, you would expect that elephants could walk across these gaps, but even with elephants, work has shown that the Palghat Gap acts as this barrier to dispersal. So this starts to give us some idea about how populations diverge from each other, how species diverge from each other, how they may do so with adaptation, without adaptation, and so on. Now, I'm at the end of my current sort of hello, hang on, let me, yeah. Section of the talk. So I'm going to give you an assignment in a minute. So if you want to have, if you would like to ask me any questions about this, which is what I've broadly presented to you at this time, is our inference of evolutionary history, of the relationships of organisms from each other, the divergence of organisms from each other, and how they and the relationship with the geography of the earth. So historical or evolutionary biogeography, where they evolved, whether with or without adaptation to the environment, and so on. So that this is sort of my broad scale summary of patterns of evolution. And we'll talk a little bit after the break about the processes of evolution. Yeah. And I'm going to give you an assignment right now. which I'd like you to carry out on pieces of paper. And again, you can keep it to yourself or send it to us. So have you guys got your pieces of paper and pens ready? We're going to take a little coffee break right now. So we'll start again at 11:40, I think. But let me see if there are any questions. If you guys have any questions now.
Hi, Karthik. There's a question from the YouTube chat.
Yeah.
So Liza Chankoti has a question. So her question is, if we are interested in randomly building phylogenetic trees among different unrelated organisms, what would be the smartest way or criterion to pick the organism groups?
You've really got me there. You want to? Well, she's on the YouTube chat, so she can't actually clarify the question. I'm not sure why you would want to build evolutionary trees between random unrelated organisms, because if you were choosing them randomly, there would not be a systematic way to choose them. So maybe I've misunderstood your question. So maybe you just want to clarify what that question is, and in the meantime, I'll take one from this chat group here, which is how to understand an evolutionary tree, a simple one and a complex one. Well, simplicity and complexity is a little, those are themselves relative terms, in the sense that I'm not sure what one would mean when we said simple versus complex. Complexity can sometimes arise from. So let's look at it. Let's look at building an evolutionary tree using 3 different methods, right? Let's build an evolutionary tree using just morphology, which you guys did when you looked at that mammalian tree. You were like, shrews look like elephant shrews, elephants look like manatees. These guys must be related. Kangaroos are very distant, right? Let's build a different one using genetics, which is the one I showed you. Elephant shrews, elephants and manatees are closely related to each other. Shrews are not, kangaroos are even more distant. Now I can build a third tree of relationships based on geography, right? Who are all found in the same geographical regions. And the question that I might want to ask myself with regard to simplicity and complexity is, do these all match each other perfectly, right? Now, when they match, in a sense, that's like, I would imagine that as simple. Okay, they look alike, they are closely related. There's nothing to fret my, poor brain about here. Complexity arises when, or the more difficult questions arise when your morphology doesn't correspond to your genetics, or your genetics doesn't correspond to your geography. Then you're like, oh wait, how did this happen? Now I have to figure something out. So that's one level of complexity, which is sort of deriving from the inherent variation in the system. Another level of complexity could arise, like when I gave you those examples of those that are there are some groups that are genetically very similar, like very not divergent at all, but morphologically they're massively different from each other. So this often happens in adaptive radiations, right? They're genetically similar, but morphologically different. In the other case, you have guys that are genetically have been diverging for years, but are morphologically similar, right? So again, I have to explain the processes by which that came about. Now, these are all real patterns in ecology. Yeah, these are this is not measurement error. This is in fact, you do see discordance between genetics, geography, and morphology. You do see discordance between rates of genetic change and rates of morphological change. This all happens, and we have to try and understand how it comes about. The other level of complexity, the other level of uncertainty rather than complexity comes about because of measurement error. Morphology and genes even genetic data doesn't always reflect the exact nature of relationships. And the tree building methods will often give you different possible sets of relationships with different degrees of confidence, and we have to understand that. So that I would say would also determine complexity. Someone's asked me to show the tree again.
Purnima has asked you to
Yes.
go back to the slide where you're talking about.
You guys see the yeah. Can you guys see it? I'm going to answer another question that I'm seeing, which is that should migration be given importance while building a tree? Well, the tree migration, we use dispersal or migration. If you migration is a particular kind of dispersal when interpreting the tree, right? The data that actually goes into building the tree might be A, genetics or B, morphological traits, or I could also, we don't generally build trees using geographical relationships, but I can also ask that question of who, which are related to each other in terms of the areas that they're found in. Right, so I can ask that question. So the tree is built on traits, whether it's genetics or morphology. I would use dispersal to interpret why certain species are found where they are, such as in this case. Purnima, do you have a question based on emphasize this a little bit more? Sure. So if you can see my pointer over here, you can see here that the group deriving from Madagascar, India, and Southeast Asia form a single group that's related to Africa and South America independently. So the group comprising Madagascar and India separates from Africa and South America first. Then a group, comprising Indian fauna separates out from that, and some of them disperse into Southeast Asia, and so you get some Indian fauna that are closely related to Southeast Asian fauna, and then you get some ancestral Indian fauna that are sort of the one over here that are sort of ancestral to that entire group, that essentially derived from that relationship with Madagascar. The alternative being the Africa and all of its relatives diverge from India and Southeast Asia, and then a group of Southeast Asian fauna disperse into India, and therefore India's closest relatives, the ancestral groups of those Indian fauna are Southeast Asian relatives. Yeah, so that's what these two scenarios are showing.
Hi, Karthik. We have a question from the YouTube chat. It's also connected to Swastik's question as well. So the question by Mikhail is, what are the morphological differences seen between the elephants on either side of the Palghat Gap, or were the difference only genetic? And I think this can be connected to Swastik's question, where they ask for the plausible reasons for the evolution differences between the clades on either side.
Sure. So as far to the best of my knowledge, there are no morphological differences between the elephants on the two sides of the Ghats. Now, the evolutionary differences between clades on different sides of the Ghats, some of it can be explained by climatic history. So during the Pleistocene, during the glaciations, much of peninsular India was covered by ice, and many habitats became uninhabitable. And there are believed to be these refugia in the southern Western Ghats, where many species survived, where forests survived. And so perhaps the forest-dwelling species only survived in the southern Western Ghats. Because remember, we don't have fossil evidence. We're basing this on our interpretation of the species we see today. The second is that the southern Western Ghats, if it historically and now has been wetter than the northern Western Ghats, then that has affected the habitats in the northern Western Ghats, which means that there would be more open habitat species such as in the example of the frogs that I gave you. But this is just one way in which the evolutionary differences can manifest, and in different groups they may have been manifested in different ways. So if you're asking why the adaptations or the morphologies of groups in the southern and the northern Western Ghats are different, that's the reason. Why are they separate from each other is because these have not these groups have not dispersed across the space in that evolutionary time. If you look at birds, apart from, in fact, I know I gave an example of the Sholokidis and laughing thrushes, but actually on the whole, if you look at birds, you won't find these differences because birds, for the most part, can actually fly quite freely across both the both sections of the Ghats. Okay, so I'm going to give you guys an assignment, two assignments actually, and so I'm going to ask you to draw two sets of trees. That is, there's an assignment 1 and an assignment 2. Each of them has two alternate scenarios, right? Here, let's take another wonderful biogeographical area, the Andaman and Nicobar Islands. This is the Andamans in the north, going from like North Andaman, South Andaman, Little Andaman. There's a Central Nicobar group, and then there's a South Andaman group. Great Nicobar is only 100 odd kilometers from Sumatra. Let us say that species colonized the Andaman Nicobar Islands by colonizing North Andamans from Myanmar, and then they slowly moved south. And that's scenario 1. Sorry. And scenario 2 is that they colonized the Andamans from Myanmar, while the Nicobars were separately colonized by species from Sumatra. Now, these should give you two different kinds of trees. And I just want you to draw these two kinds of trees for yourself based on your understanding of whatever I described earlier. And what would these two branching trees look like, which depicts the closest relationships between those groups. So the tips of your trees over here would have Myanmar, Andamans, Nicobar, and either Sumatra or Southeast Asia. Okay, now that's one homework. The second one is let's look at a human ape phylogeny. And I don't want you to worry about which is the right one, but I'm giving you some scenarios again, which are that humans are most closely to a group which comprises chimps and gorillas. That is, the chimps and gorillas are really closely related to each other. And the second scenario is humans are most closely related to chimps, followed by gorillas, and then by other primates. I've given one example here, which in fact does not depict either of these scenarios. And but then again, if you were basing it entirely on morphology, you might actually say that this phylogeny is true, based well, not on morphology, based on facial expressions. You might say that this is the correct version, but it is not. Regardless, let us try out those two scenarios. I am going to take a break for coffee, which I need. I will give you 10 minutes to do this. Let's say 15 minutes. Let's start again at 11:50, and I will see you all back here at 11:50. In the meantime, if more of you have questions, you can put those questions in the chat box, and I will take those questions as soon as I come back.
Yeah, but
Right?
no need to log out. Just stay on here. Okay, just mute
Everybody can
yourself.
stay on. We're going to put our videos off, but we'll
because
be back.
admitting people will take a lot of time otherwise if we leave now. Okay.
Yeah. No need to log off. You can stay on. Just you've got your videos and audios off. That's great. Just like scribble with pencil, paper, have coffee, tea, buttermilk, whatever it is, and we'll do a phylogeny of beverages when we come back.
Okay, great.
In India, endangered and migratory birds are included in the Schedule 1 of Wildlife Protection Act of 1970.
Okay, it's 11:50, and I perhaps a few people have not come back yet, but I think what I'll do is I'll look at a few questions in the chat box while we're waiting for everyone to grab their coffee and come back.
Hi, Karthik. Can I ask a question from the chat box if no one has a question right now?
Sure. Yes. From the Zoom chat box, Gokul?
No, this is from the YouTube chat box. This has been.
Okay. From the YouTube chat. Oh, good, great. Let me just take the one from this one, and then we can do the YouTube. Yeah.
Sure.
Adding on the point of dispersal, Purnima, I think, says, my question is, how does diversification at a species level play a similar role, if any? Diversification. So one of the points to note here is that the different tools that we use, like let's say we're talking about genetic data, the genetic data can show us diversification at different scales. So you have to use some kinds of genetic markers to, for example, tell the difference between individuals. Other kinds may tell you the difference between populations. Other more genes or markers that evolve at a more slow rate may be able to tell you the difference between different species or different genera or families and so on. So the rate of change in that gene itself will make it amenable to telling you the differences at different scales, going from individuals to populations to species to higher levels of organization, right? So if I'm primarily focusing on looking at differences between species, or between, let's say, between genera, because I just want to know broad scale relationships, that may not tell me the difference between species. If I use a finer resolution genetic marker, that potentially could tell me the difference between two closely related species, and an even more fine one might tell me differences within species, saying, okay, this population, from Western Ghats is different from this population in the Eastern Ghats or in Southeast Asia, even though they're closely enough related for me to call them the same species. Now, one of the things that I was not going to talk about today, or decided not to spend too much time on it, was what is actually a species itself. And the answer to that is fuzzy, in the sense that there are many species concepts that have been posed over the years, and there are many different views on whether we should use biological sort of criterion of reproductive isolation, that is groups that don't interbreed with other groups, which was proposed by Ernst Mayr, or evolutionary relationships, or morphological similarity. So there's lots of diverse opinion on species concepts, and I'm not going to get into it today. And I'm making that point simply because as we come down this tree and I start making it finer resolution, remember I said that within a species, a population in Western Ghats and Eastern Ghats may be different. Who's to say whether they're the same species or not? There is no straightforward answer to that, right? But as we use finer and finer genetic markers, we can sort of understand those relationships at finer levels of organization. So going from families, genera, then species, then populations, and finally, of course, even differences between individuals. I hope that answers that question. Gokul, go ahead.
Hi, this is a question from Rudranath. His question is, since South America is also a tropical rainforest region, why aren't species like elephants, king cobras, etc., not found in these regions, whereas these species are found in Asian tropical rainforests and Africa?
Dispersal and the history of the earth, right? Which is why I spent maybe not enough time, but talking about how continents were connected to each other, and that's how dispersal took place for the greater part, right? Some species of of plants and animals have been able to make transoceanic journeys, but many of them have dispersed over land, right? And so elephants crossed over the land connections and made their way into India. King cobras likely originated in, well, we don't know exactly where they originated, but they originated in Southeast Asia and spread through Southeast Asia and into India because those land masses were connected, right? So there is a, when you study the patterns of diversity in the world, there is one rule called everything is everywhere and the environment selects, right? Which basically means that all species had the potential to be everywhere on the planet, but where we find them today is simply where the conditions are suitable and not, right? And maybe this is true for like a even it's not completely true even for the best dispersers in the world, such as wind dispersed plants and birds and birds with great migratory ranges. Even for them, it's not true. But you do find some birds that have like a pan-global distribution and plants as well. But that's fairly rare. Most plants and animals, we find, as I said, where they originated, where they were able to disperse to, and not disperse like in a matter of few years, but in evolutionary time over millions of years. And then if they were able to survive there, right? So those three processes are important. And where they could get to depended on how they could get there. And if they were terrestrial, it would have depended on there being land connections at a given point in time. I hope that answers your question, Rudranath. So before we go on to the next part of this session, which is about evolutionary processes, I just want to share with you the some of the possible results that we might get from those scenarios. Let me share. Which screen is it in now? Yeah, there it is. So we can build a human evolutionary tree in different ways. In fact, here 3 examples are given. The tree A represents, I think the first scenario that I gave you, where chimpanzees and gorillas are sister species. I haven't used that term before, I think, but sister species are essentially pairs of species that are most closely related to each other relative to everybody else. So chimpanzees and gorillas are sisters here. Those are most closely related to humans and then to other primates. Scenario 2, which I gave you, was chimpanzees and humans are most closely related to each other, thereon to gorillas and others. And there's a scenario 3 given here as well. As a matter of fact, tree B is the correct one. Humans and chimps are in fact most closely related, and then to gorillas, and within chimps, of course, there are chimpanzees and bonobos. The other scenarios I gave you, again, I'm going to give you fairly like simple versions of these. If the Nicobar and Andaman were colonized independently by Myanmar and Nicobar on, the 2 cases, you would, in your tree, you would see that the closest relatives of Andamans are Myanmar species and Nicobar are Sumatran species, all of which derived from Southeast Asian, presumably. But if they derived only from Myanmar and then colonized through the Andaman Nicobar Islands, and I'm not talking about any particular path of colonization over here, then both of that group comprising Andaman and Nicobar species would be most closely related to Myanmar species, and all of them to Southeast Asian species. Yeah, so that's just sort of answers to your puzzles. Okay, shall we go on? Are there other, let me see if there are questions that
There are no more questions in the Zoom chat.
Okay.
There's a question in the YouTube chat, which I think can be answered after your talk in the next section that you're following up, which is connected to adaptation and evolution.
All right, cool. All right, let me just bring up the. So one part of what we're interested in are these large-scale patterns, and we do make inferences about evolutionary processes, such as uh, the rates of diversification, that is the rate at which new species arise, um, and about dispersal and so on, right? But if we want to dig down one more level, we want to understand these processes themselves as to, how does this come about? And this also can be divided in terms of looking at it at a slightly larger scale in terms of speciation and extinction, and a slightly more, and at the genetic scale itself, is how do these how are these explained at the level of genetic variation, right? So to start at the larger level, uh, we've repeatedly talked about diversification, right? And diversification, in a sense, can be considered as a combination of the two processes of speciation and extinction. So new species come about, some go extinct for a variety of reasons, and these processes, in a sense, result in the diversification of a particular group. And I so showed you many patterns of diversification. Now, let's say I said that there are two examples where the fauna of the Western Ghats may have been derived from the fauna of Gondwana, or it may have been derived from the fauna of Southeast Asia. Now, if we simplify that, we can start with the idea of an ancestral species that then splits into one or more species, right? And the simplest formulation of that, an ancestral species splits into two species. Now, this can come about in broadly three different ways, which we call allopatric, sympatric, and parapatric. Now, allopatric speciation, essentially two populations are separated from each other geographically and eventually diverge over time. There may or may not be any morphological differentiation between the two. Sometimes, in fact, when you have like with many frogs in the Western Ghats, when you have frogs on mountain tops, because the conditions are similar to each other, they have diverged because they're geographically separated, but the conditions are quite similar, and so they may not diverge morphologically. The second comes about when they are in the same place. So there is no geographical isolation, but there are other mechanisms by which speciation can take place, does take place. And the third is called parapatric, which is essentially along an environmental gradient, for example, here depicted as a temperature gradient going from the bottom of a hill to the top of a hill mountain. So you have high temperatures to low temperatures, and along this, for reasons driven by this environmental gradient, the populations start diverging from each other. And this is known as parapatric speciation. So these are the three broad modes of speciation that are described in nature. Now, allopatric speciation is of particular interest because for a very large number of groups of animals in particular, the dominant mode of speciation has been found to be allopatric speciation. And through history, with many taxa, it has been found that this reproductive isolation, or this isolation of populations from each other where there is no gene flow and the gene flow is cut off, then it results in allopatric speciation. But allopatric speciation can itself be thought of in two particular ways. One is when there is a large ancestral an ancestral population of some size, right? And there is some barrier in between. Now, remember, right? Like we gave so many examples of these barriers forming because continents moved apart. But those barriers could come about in other ways. Those barriers could have been due to the formation of a gap like the Palghat Gap. It could be because a new river was found, or a mountain chain was uplifted. So when the barrier is formed and it separates two populations, this is known as vicariance. Again, the term is not important, but this is a particular kind of allopatric speciation where the formation of this barrier in a pre-existing population and preventing therefore gene flow between now parts A and B of that results in these two diverging from each other and becoming two different species. The other, which is, it's called dispersal. Usually when we say, allopatric speciation, one type versus another type, it's, people will usually use the terms dispersal versus vicariance. Here what happens is that the more important part is that there is sort of what is called a founder event. So you have an ancestral population. Now this population goes and settles somewhere else in a new location. Now in the ancestral location, it continues to accumulate changes over time, but the population that has dispersed, usually to a distant location, so that there is no gene flow now between these two bits. This dispersal event, that population now starts diverging and becomes a different species. Typically in founder events, they tend to be a few individuals that have dispersed, say, to an island or something like that, and have founded a population that diverges into a new species. Very often these are based on just a few individuals. Whereas in a vicariant event, you could very well be dividing two populations that are both equally large, or in some cases it may be that one is large and one is small. Now, once populations re-establish contact, if that should happen, enough time or enough change has accumulated that they may not interbreed or produce fertile offspring, right? They may interbreed freely and extensively, or hybrids may be less fit. Now, if hybrids are less fit, there are many species. In fact, in contact zones, it is known that they produce hybrids, even in mammals, for example, with Nilgiri langurs and common langurs and so on, which are, any primatologist would say these are different species. They have different, evolutionary genetic histories. They're morphologically different. They have different calls, but they do interbreed in contact zones. And we don't know if the hybrids are less fit, but one way that these populations are kept separate is that the hybrids are less fit. If they interbreed freely and extensively, then that population will not diverge into two species. Of course, if they are unable to breed at all or do not produce fertile offspring, then these populations' genetic histories will in future remain independent of each other. It's a little bit less common, but the other form is sympatric speciation, which can be caused by a number of processes again, such as disruptive selection, chromosomal changes, which happens very frequently in plants, chromosomal rearrangements, and so on. And there are several examples of this, of speciation in isolated lakes, involving with cichlids and sticklebacks and so on, where these changes have essentially taken place in the same geographical region through mutational changes that have come about through various mechanisms, but those causing adaptation to different different habitats, such as different zones of the lake, or as the case may be. I'm not going to talk too much about parapatric speciation, because in some ways the mechanisms are actually would either come about by the mechanisms of sympatric speciation through disruptive selection, or they come about through the mechanisms of isolation in allopatric speciation. So the mechanisms are not per se different, but it takes place along an environmental gradient. If we talk about speciation, we have to talk about extinction as well. Now, extinction is believed to come about again through for a variety of ecological processes. Most famously, there is the what is proposed as the Red Queen hypothesis, which the Red Queen in Alice in Wonderland says takes all the running you can do in the same place. So species are constantly having to adapt to a changing environment. or to the biotic environment in terms of competition from other intraspecific competition, of course, but then interspecific competition, which is competition with other species, predation, and parasitism, and so on. So that all species are in some sense also believed to be in an evolutionary race, and that evolutionary and you have to keep up with the race. And if you're unable to adapt to changes in the environment, or adapt to changes that your the biotic environment, then there is, of course, a probability of becoming extinct. Now, broad patterns have been found also in rates of extinction when you're looking at it in a sort of in a macro evolutionary sense. That is, the probability of the species becoming extinct has been found to be independent of its evolutionary age. But certain taxonomic and ecological groups have higher rates of extinction. So if for example, if you compare small versus large, herbivorous versus carnivorous, and so on, they've been found to have different rates of extinction. But much of this, but while we talk about the ecological factors which might drive extinction, one must also remember that there are chance events, what we call stochasticity, and the stochasticity can arise from genetic variation. It can arise from environmental variation, including catastrophic events. There might be, if you may be on an island and you might be hit by a hurricane or a cyclone or an earthquake, or it could be demographic events. I mean, which is like in small populations in particular, by chance, if there are, let's say far too many males and not enough females, that will affect the rate of population growth, and this can lead to the extinction of small populations. So chance variation in population factors, environmental factors, just many years of a drought, for example, or in genetic variation and mutations could also result in extinction. And one can't talk about extinction without sort of mentioning that there have been these amazing diversification events in history. There's the Cambrian explosion of life, the advent of dinosaurs, the mammalian radiations, and so on. But there have equally been some dramatic extinctions. The Permian extinction at the border of the Paleozoic and Mesozoic era, about 250 odd million years ago, resulted in the extinction of, I think, 90% of terrestrial and 70% of marine fauna, or the other way around. The Cretaceous tertiary extinctions, which resulted in the disappearance of dinosaurs, which is attributed to an asteroid impact, much talked about, and the disappearance of many mammals during the Pleistocene, which is in fact attributed to the spread of early humans across the planet. So I'm going to come back to this briefly. We have speciation and extinction, which constitute the diversification, but then we also have this process of dispersal. And quite remarkably, more than 2,000 years ago, Aristotle himself said that we must understand the meaning of notion, for without it, the meaning of nature itself would be unknown. So in a sense, the idea of motion and change which underlies dispersal or movement of animals is one of the most remarkable processes that results in life on Earth as we see it today. Now, if you think about dispersal itself, what is dispersal? Is the mobility of plants and animals. It's important for us to make the distinction between dispersal as an ecological process, that animals will move away because of reduced plants as well, to reduce competition with parents and siblings, at the cost of habitats becoming dissimilar. So, plants disperse, right? Their seeds disperse and go far away from the parent. Tiger cubs disperse. Various animals disperse. But this is different from dispersal as an evolutionary process, which results in these large-scale changes in geographic distribution that we see today. But These may be rare events and they may be infrequent and stochastic. But over hundreds of thousands of years, they result in the changes that we see in our world. And to define the process itself, in order for a species to expand its range or move to it must travel to an new area, withstand potentially unfavorable conditions, establish viable populations upon dispersal. And this can happen in 3 different ways, which are called, let's just call it jump, dispersal, and diffusion for now, which is that it can happen in one sudden jump, or it can happen gradually over time. So that's sort of 2 broad classes in which this can take place. And I also wanted to emphasize that barriers need not just be physical. Those are the ones that are easy for us to think about, mountain, river, sea. Why do we find so few frogs on islands? Because they're generally like not tolerant to seawater, to salt water, and so it poses a massive physiological barrier for them, right? Physical and physiological barrier for them. But just physiological barriers themselves could be in the form of physical tolerance limits, environmental temperature regimes. All of these could pose barriers as well. And they can also be ecological barriers in terms of competition and predation, behavioral and psychological barriers and habitat selection and so on. And in fact, if you think about the animals found about marine organisms, you would think, oh, unlike land organisms, why aren't these found all over the world? I mean, like they should actually have like really large ranges. And it's actually true that many marine organisms have huge ranges because they can actually disperse across the ocean. However, there are also temperature and salinity and ocean current-based barriers that actually prevent organisms from dispersing wherever they want, even in oceans, resulting in geographical isolation, which then, of course, results in divergence. Yeah. I already talked about this, that dispersal is one part of the problem, but the other is actually like establishing yourself over there. And there's a number of factors which affect survival once you've arrived in a new location. And here again, your ability to adapt to that environment will influence your ability to survive there. But that process also leads to a form of leads to divergence or change, which we then see in the formation of new lineages or species. Okay. All right. So I'm going to get into the last part, which is sort of looking at the evolutionary processes in terms of genes and alleles. And I will give you an assignment at the end of that as well. But in the meantime, if there are any questions, I can stop now and take a few questions.
Hi, Karthik. There's a question from Shruti. She asks about can you explain about psychological barriers? Psychological.
Yeah, I can see that. Yeah, so well, it might seem like only humans have sort of psychological issues, but when I say psychological, these could be that's also sort of that's also behavior, but is behavior relating to, so for example, an innate fear response, right? So there's work these days on what's called landscapes of fear. So prey, for example, will or will not forage in certain areas because of the presence of predators over there, right? So their movements are determined by the presence of predators. And this has been shown in both terrestrial and marine systems. And those sorts of the behavioral responses might themselves pose a barrier, right? So if I'm just to give like some small scale example, I might be in two So you know, in the higher altitudes of the Western Ghats, you get these shola grasslands, which are like these little patches of forest surrounded by grassland. Now, if I lived in a shola patch, in order to get to another shola patch, I have to go across a grassland, right? And the probability of predation might be much higher over there, and therefore I might be behaviorally adapted to avoiding it, right? And so I do not cross that barrier for a behavior. In this case, one might call it the behavioral response being itself being sort of the psychological response to fear of predation, right? So that's one possible example of a psychological barrier.
Hi, Karthik. We have a question from YouTube chat.
Sure.
So this is from Rudranath. So his question is, since birds can fly over, and as well you do know about migratory birds, how does geographical barriers make differences in birds and speciation?
So it's a good question. I mean, in the sense that if you look broadly at groups, if you compare birds and frogs, for example, right, you will find that on the average, frogs tend to have much smaller ranges. Birds tend to have larger ranges, and in fact, marine organisms, some marine groups have even larger global ranges because their ability to disperse, the average ability of species in that group as a whole is much greater. That's what it sort of implies. Yes, because birds can fly, marine organisms can sort of like disperse passively through the water. Frogs are just not that, jumping only gets you that far, right? However, within those groups, the ability of an individual species to disperse may be much lower, right? So ground-dwelling birds, like the laughing thrushes, of which I gave you an example, may have lower dispersal ability to begin with, and for a combination of their adaptations to certain habitats and so on, may not disperse to the degree that other birds do, right? And so you end up getting a pattern for those, the Sholicolas or the Sholicola, the genus, and for laughing thrushes that are actually quite similar to frogs. So it looks like they are only dispersing as much as the frogs are dispersing. So that's how you end up getting those divergences, because you've had the same sort of isolation for those species as you've had for organisms like frogs that don't disperse much at all. I hope that sort of answers that question. And I particularly like to give the example of plants. People will say plants and like do studies of like entire plant group, right? All woody plant species, 500 species across 20 families, or not just families, but like orders. And the remarkable thing is that when you're looking at a group like plants, where actually it's almost as if we're looking at all, much broader than even like all, vertebrates, right? Vertebrates, invertebrates, all of it. And so you can have plants that behave like birds that disperse very, very widely, and then you have plants that behave like frogs that don't disperse at all. So you can get the entire range of dispersal strategy when you look at plant groups. You can ask why don't species disperse more? Well, there are costs to dispersal. There are energetic costs. There are survival costs. So the particular individual has to trade off the benefits of dispersal versus staying in the same place, right? And so those are the costs. Those are the trade-offs which determine sometimes whether species end up dispersing or not. Of course, also depending on physiological constraints. If I'm a frog, I inherited certain body, morphology. If I'm a bird, I inherited wings, most likely, that gives me a certain ability to disperse or not. But within that, I still have to make decisions about my own survival when I make a decision to disperse or not. There's a question about there's a giant lizard in the Galapagos Islands now. There is a giant tortoise, and I actually, yeah, I don't know the answer to the lizard that was spotted in Mexico. I haven't been up on that, but there are giant tortoises in the Galapagos, and the giant tortoise of Galapagos has its closest relative in the Aldabra Islands in the Indian Ocean. There's only 3 species of giant tortoises left. There are also giant lizards on islands such as the Komodo dragon or the Komodo lizard, which is a species of Varanus or monitor, and they have found in particularly many groups of animals, but mammals in particular, and certainly like with reptiles as well, such as with the giant turtles and lizards, that on islands you tend to get increase in body size for smaller bodied animals, and this might be because of the absence of predators and so on, but you also get reduction in size for larger bodied animals, and that might be because of the absence of sufficient resources. So for example, elephants on islands have been found to be smaller. The other species of large mammals like rhinos, and there have been many examples of large mammals that became smaller on islands, and many examples of small mammals that became larger on islands, which is believed to, which of course is like the changes take place over time, but is believed to be driven by a combination of biotic factors such as competition and predation, and abiotic factors such as availability of resources. Okay.
Are you open for one more question?
Sure, I'll take one more.
So this is a question by Mikhail. His question is, would speciation along different zones of a canopy count as sympatric or allopatric speciation?
This is a very good question, actually. Not just around the different zones of a canopy. There are we actually have examples of frogs on the Western Ghats where from the same mountaintop, where one species diverged on the grassland from its relative, which is in the forest, and at one scale people would call it sympatric, but on the other hand, you can say that they were just like, actually there was some kind of a habitat barrier between the two. So my question actually would be that I would base that on understanding whether that difference came about because they got isolated from each other by just physical isolation, in which case I would call it allopatric, or if there were other mechanisms that caused that divergence to take place, such as genetic mechanisms that led some mutations that caused them to adapt to different microhabitats, disruptive selection, or other chromosomal mechanisms that actually then prevented interbreeding, right? So to me, just they're the same location. If it's just selection along a gradient, people might call it parapatric, and might call it different terms. So I try not to get the terms apply to certain processes, but the processes underlying it are more important to understand. So what actually happened here is really the question that I would ask. Did it just so happen that for some reason these guys never came into contact with each other? Like if they're all living on the canopy of all trees in a forest, I find that hard to believe that there would not be some contact. And so it might actually be because of some other mechanism. But on the other hand, it could, even a small amount of, barriers that prevent any gene flow between the groups could also result in that divergence. So I'll toss that back and say the answer would there is no yes or no answer to that. The answer would be in understanding the mechanism that led to speciation. All right. It's 12:30 and I'm going to turn into a pumpkin or something. So at 1. So before that happens, let me go through the last part of today's session. And I'm not going to dwell too much on this, but it's something that you would come across quite well. It's a very fundamental part of understanding understanding evolution. In fact, if you ask, and many evolutionary biologists. Sorry, let me just stop sharing that for a minute. If you ask many evolutionary biologists, what is evolution? At a broad level, people might say descent with modification, change over time, and so on. Very often, people will say evolution is a change in allele frequencies. What is an allele? An allele is a copy of a gene, and you'll have many alleles for a particular at a single locus or for a particular gene. A common example being the blood groups in humans. So at the ABO locus, you have different alleles such as A and B and O, and this is true for all loci or for all genes. And changes in these alleles, these allele frequencies, really underlie the process of divergence of population A from population B. So when we talk about evolution at that scale, we're really talking about divergence between populations and not necessarily species, because at the first level you have these little units which we call populations, within which gene flow occurs, and then they're isolated from other populations, and some amount of gene flow may occur between them. It may be a lot, it may be a little, and then when gene flow stops completely over a long period of time, we say, oh, magic, we got a new species, right? But like that process, the process at which it initiates is really the processes that happen at the genetic level within this population, right? And that is defined for a particular trait by the allele frequencies. So what is the proportion of A's in your population, what's the proportion of B's in your population, and therefore if A and B are two alleles, you could get an AB, and so on. So we're just going to very briefly look at the processes that underlie that. A very large part of evolutionary biology studies, in fact, devoted to understanding these changes in allele frequencies, understanding these changes at the genetic level, at the population level. This is done both through studies in nature as well as through experiments. There's a huge body of work in experimental evolution that I'm not going to talk about. It has involved model organisms such as flub beetles, Stribolium or Drosophila, and so on. And I'm not getting into any of that today, but I just wanted to flag that that's an entire sort of field of study by itself. Much of what I focused on today really has been on the broad scale patterns and processes of inferring evolutionary history. So let me just briefly touch upon the idea of gene frequencies. And let me share my screen again. And also because this is something that you're very likely to be, to be asked about at, almost any interview that you go to, and what I covered today may not give you enough to deal with it, but I wanted to flag it because it's something that you'll want to familiarize yourself with. Now, Hardy and Weinberg came up with the idea that genotype frequencies in a population, in a large population with random mating, will remain constant from generation to generation in the absence of other evolutionary influences, right? And these other evolutionary influences there are several that could result in changes to genotype frequencies, but the main ones are, as you can see here, no selection, which means that there is no preference for type A or type B, or for the dominant allele A or recessive allele A. There is no preference for the phenotypes that the genotypes result in. The second is no mutation, so no weird three-eyed, I was going to assume these are frogs. No migration, which means that like that river, that barrier that I told you about is, you know, a complete barrier. The frogs don't like to swim across it. Let's assume I put a whole huge amount of salt in it, so they're all like different from each other. They're separated from each other. The population is really large, as I mentioned before, and mating again is random. So there's no particular pattern there's no mate choice or preference in mating, right? These are the assumptions of Hardy-Weinberg equilibrium. And for example, if you look at one of those population, which is the size of the population, it has been shown, this is just a sort of a simulation model, that these changes in these allele frequencies, which is shown on your y-axis over here, is dramatically different in small populations. This is just a simulation. It's just like some example of a population size of 10, and you just like randomly, change, you have, you model for that population to produce multiple generations. And over those generations, you find a lot of this variation in allele frequencies, which you do not find in large populations. And this is because of chance or stochasticity. What does the Hardy-Weinberg principle actually state? If you have two alleles, let's say big A and small a, it says that given random mating, that the frequency of those alleles in the population, let's say that the proportion of allele A is P, the frequency of allele Q, small a is Q. So let me repeat that. I might be going a bit fast. Let's take big A and small a. I'll show you an example in a second. P is the frequency of capital A, Q is the frequency of small a, P plus Q is equal to 1. That the frequency of the homozygous dominant genotype, which is capital A will be P squared. The frequency of the heterozygous genotype will be 2pq, and the frequency of the homozygous recessive genotype will be q squared. So capital A is P squared. Capital A small a, which is the heterozygous genotype, the frequency of that is 2 into P into Q, and the frequency of the homozygous recessive genotype is Q squared. Sorry, this is a little blurry. But if I work that out with an example, now, if I say that the frequency of P is 0.3, then the frequency of Q has to be 1 minus 0.3, which is 0.7, and that will give me the, in Hardy-Weinberg equilibrium, my expected genotypes of capital A, small a, and small a can be calculated based on the equation I showed you before, which is, or is at the top of this slide. So the frequency of capital A is P squared, which is 0.3 into 0.3, which is 0.09. The frequency of capital A small a is 2pq, which is 2 into 0.3 into 0.7, which is 0.42. And the frequency of Q squared is, frequency of small a is Q squared, which is 0.7 into 0.7, which is 0.49. Okay, so broadly, this is what Hardy-Weinberg equilibrium states. And so I can understand also, supposing my frequency of P and Q were 0.3 and 0.7, but what I actually find is that that the proportions are not in the, or the frequencies are not in the proportions that I expect. I can then come to the inference that there is some evolutionary process at play here. I might not know which one. So what we see when we talk about how evolution actually occurs, it occurs by a series of processes. We focused a lot on natural selection, resulting in, either in in adaptation to the environment, or coping with with an arms race with a competitor or a predator. But it can actually come about due to any of these processes that result in changes in allele frequency, right? So it could be natural selection or drift or mutation or migration. Any of these could result, and a combination of these results in changes across generation, which results in adaptation and modification. As we've said, the consequence, which it could be adaptation, it could be modification, that is not necessarily adaptation because it has come about because of the processes of genetic drift, which is chance changes in small populations resulting from, say, mutation or recombination, and not necessarily selection, which to abiotic or biotic factors, right? So, as I said, I'm not going to go into this in too much more detail. I think that's about it in terms of slides, if I'm not mistaken. I did want to give you, yes, I just wanted to thank a bunch of people for slides and sources and so on, which are cited elsewhere. But I want to go back to this for a second and give you a couple of assignments to close this out. Let me unshare that so that I can see my screen. So I'll take questions at the end of this, which can cover, either the last section or any part of today's session. But before we do that, let's spend a couple of minutes doing two things, two exercises. Exercise number one is I'm sorry, I haven't written these questions down, so you'll have to do that yourself. Question number one is, what are the, under Heidi-Weinberg equilibrium, what are the expected frequencies of genotypes if P is equal to 0.6 and Q is equal to 0.4? 1B is, at what frequencies of P and Q would you expect to see the highest proportion of heterozygotes in the population? So work that out for yourselves, and don't put it in the chat box because everybody's working on it. And I'm going to give you a couple of minutes before I start taking questions. Oh yeah, I had to give you a question too, right? Yeah, guys, don't put your answers in the chat box. Yes, it's too late for that, but apology accepted. So can you guys work out the following? So two things I actually want you to do at home. Now, the question you have to ask yourself is, what is this? How does this work, right? Now I've got this Hardy-Weinberg equilibrium thing, Hardy and Weinberg said, and then Dr. Shankar repeated that, like, if the proportion is P and Q of the two alleles, then P square plus 2PQ plus 2PQ and Q square define the genotype frequencies of the different genotypes. But how is this maintained over time? Why should it always be constant? Now, what you can do is that now that you've calculated in principle, and this is homework, you can look at in generation 1, if I had, my proportions of P and Q were, whatever those values were, and you don't need values for that. Actually, you can do it without actual values. Now that resulted in a particular set of genotypes, right? P square plus 2PQ plus Q square. And so now I know the proportions of A small a, and small a. Now for that generation, for the next generation, the genotype frequencies are going to be dependent on the proportion of P and Q in generation 1, which I've just produced. No? So calculate for yourself what is P and Q for this for F1, for generation 1, which I've just now calculated for. Now I have the genotype frequencies. Now calculate back the values of P and Q from that and see what you get. Okay. And there's no surprise to say that you would expect P and Q to be the original P and Q. That's how this is maintained over time. However, make that calculation to assure yourself that it is true, because then you will have understood how this works. A similar problem that I can now give you is that supposing I find that the genotype frequencies capital A is 0.4, capital A small a is 0.2, small a is 0.4. Those are the genotype frequencies, not the allele frequencies. Remember, capital A is small 0.4, capital A small a is 0.2, and small a is 0.4. Now from that, can I calculate? I have to calculate what my proportions of P and Q are, and then I can calculate what the proportions would be if it were in Hardy-Weinberg equilibrium, and determine whether what I have given you is or is not. Right? So again, just the kind of assignments that the kind of questions that you will come across. So don't put your answer in the chat boxes. Just try and work these out for yourself. Again, I'll repeat that. The question 2A was work out the allele frequencies in successive generations of successive generations. Just 2 successive generations. You had some allele frequencies P and Q. They resulted in P square plus 2PQ plus Q square. That A small a and small a. Now, what is the P and Q of that? And if I were to give you an example where that A small a and small a is 0.4, 0.2, 0.4, then what are the allele frequencies P and Q? And is my example that I've given you, is it in Hardy-Weinberg equilibrium or not? That's a question that you can attempt to answer. Okay, so I'm going to spend the last 5 to 10 minutes that we have in answering questions. And Gokul, I see one in this chat box which says, what leads to peripatric speciation? I have to confess one thing, guys. I'm terrible at terminology. And I remember it as far as I need to. If I'm not, I could Google it, and you could Google it, and and tell me if I'm wrong. But I think it's a form of allopatric speciation that occurs at range boundaries. So it's just like at the isolated boundary of a range, there's like a population that sort of diverges and becomes a different species. It's kind of similar to parapatric allopatric speciation in the sense that they get isolated somehow, but it's sort of not like split due to a barrier, or perhaps even an environmental gradient. I'm not really very sure of examples of it either. Yeah, but that I think is what I think peripatric is. Again, I will repeat that it's the mechanisms that underlie speciation that are more important, and not the names that we give it. And to emphasize that again, was it geographical isolation? Was it an environmental gradient? Was it a chromosomal mechanism? Those are the questions that I would be seeking answers to. Gokul, are there questions on the YouTube channel?
Yeah, so we have a question on the YouTube channel. This is from Santosh. His question is, how does behavioral ecology help in plant speciation?
How does behavioral ecology, I don't know if you would consider, dispersal is a life history trait, so I wouldn't necessarily call it behavior, but that's a good question. I don't have a quick answer to that, but there might be plants might have certain adaptations to particular environments, which include anti-predator responses and behavior and so on, which might make them better adapted to certain geographical locations and areas than to others, right? Therefore, then exposure to new environments might make some populations or some populations that undergo some changes better or worse adapted to those. That's sort of the obvious one. The other, of course, is reproductive mechanisms. So plants that are monoecious versus dioecious, these may all influence both the ability to colonize as well as survive, as well as may change their susceptibility to different environmental events, to demographic events, and so on. Sorry, that's a pretty vague answer, but that's the best I've got for you off the top of my head. Does acclimatization have any effect on dispersal ability? The way I understand acclimatization, it's more. It is adapt. Well, let me put it the following way: that plants or animals that have a wider tolerance to environmental conditions will be able to disperse across areas that have those conditions. So, in fact, there is a theory that species in higher latitudes have larger ranges, and that is because the theory, and it's not always been shown to be true, is that in temperate areas, the diurnal and even the diurnal and annual range in say temperature is so high that they in order to survive at all, they have to be adapted to that wide range of temperatures, which allows them to then disperse through and disperse to areas that have that range of temperatures. Whereas a tropical species is subjected to a very narrow range and doesn't really have to be adapted to anything beyond it anyway. And so then it prevents it from actually being able to disperse to areas that do not have that range of conditions to which it is adapted. So yes, if I look at it broadly in terms of environmental tolerance, the answer is it affects the consequence of its dispersal, but I'm not sure. I mean, when we talk about dispersal ability, you think of it as a life history trait in terms of physically and physiologically how far can you go? It's like how far can you fly, or how far can you be carried? So that's what we tend to think of as dispersal ability rather than dispersal success.
We did have Dr. Maria Taka raised a hand, so I think she
answer that question. Hopefully, yes. I was going to say, may your questions shall be answered tomorrow by Dr. Thakur in the behavioral ecology session. Dr. Thakur, would you like to say something?
Will you take one more question from the YouTube chat?
Sure, I can take.
So this is a question from Ashirwad. So his question is, how does mimicry occur in organisms? Is it hit and run, hit and trial, or does it? Sorry.
Yeah. So this is true of. I mean, when you ask that question, it's true of all manner of adaptation, right? Like this is at a macro level. This has been asked about many things, like how did the eye evolve? Is a famous question. Is like half an eye is no use to an organism. So it's like how did this like complex organ like the eye evolve from nothing? However, there are obviously in most cases there is selection for there are benefits, let me say, for intermediary stages, or possibly those are neutral, that those can all occur equally in a population until you get a variant that has a significant benefit. So it might encompass a normal range of variation that is also not being negatively selected. So that's sort of to answer the question about the hit and miss. I mean, in the sense that like you're not expecting that all examples of mimicry came about because in a related organism it dramatically suddenly developed a mutant that looked like something else. But to answer the broader question of why it occurs, it could occur for a variety of reasons. There are different kinds of mimicry. Sometimes palatable organisms, not organizations, or non-poisonous organisms might mimic poisonous or unpalatable ones, because the predators have learned that those are not to be consumed, and then by mimicry these get protection. Yeah, that's one common example. Of course, often poisonous or unpalatable organizations, they mimic each other also, so that sort of like, because then with more individuals that look like that, the avoidance by predators becomes is selected for, and those organisms have a higher chance of survival. Can you explain more about the kind of evolutionary forces and the amount of deviation from divided by equilibrium? So these you can add parameters to this. So in a sense that you can say that one example that I'll give you is that that I can say that when I look at the genotypes of homozygotes and heterozygotes, I can hypothesize that homozygotes have a selective advantage, right? The dominant phenotype of the dominant allele, right? Now, what does this mean? This means that the capital A and capital A small a are being selected for, and you can write an equation for this, and you can put in a selection coefficient and all of that. But like just in verbal terms, if you hypothesize that the dominant genotype, that is like what is represented by capital A, has a selective advantage, then both AA and A small a will be selected for, because they're both showing that trait, right? And so then the A's and AA's remain in the population, A small a remain in the population, and a certain number of small a will persist. If I hypothesize the opposite, if I hypothesize that small a has a selective advantage, then the small a the phenotypes, those individuals always get selected for over the either capital A or capital A small a. right? So eventually, that allele will get fixed in the population depending on the number of generations and the amount of selective advantage. Yeah. So and then of course I could also hypothesize that the heterozygote has the greatest benefit. It's called the heterozygote fitness, and again, that will not result in either allele disappearing, because the heterozygotes have eventually the small a capital A will result in some homozygotes as well. So selection pressure or different selection coefficients is applied to different ones of the genotypes will have different consequences. Yeah. So, but we can understand exactly what the expected values will be by putting those selection coefficients in, and your original, proportion of alleles and so on. How do we understand the theory of island biogeography? An independent species is an event or an event capable of undergoing the evolutionary processes. Okay, that's a complicated question. I'm just going to answer the first part of that. I mean, I'm very briefly. Island biogeography actually, in its simplest form, did not invoke speciation. It did invoke speciation as one of the larger processes within the system, but largely what it talked about was rates of colonization and extinction on islands based on how near or far they are from the mainland, which determines the equilibrium number of species on the island. And then yeah, I'm sorry, I didn't fully understand that. I think that's Purnima. If you want to clarify that, I can try and answer it. So can you mention the characteristics of the Palgard gap with regard to isolation, no connectivity? Well, it's just the Palgard gap is like in the mountain chain, it's formed by erosion. It's a valley that comes down to about 140 meters, I think, right? And several kilometers across. And to be honest, I don't think that any of us, even when some of these studies were done 20, 25 years ago on elephants and so on, were actually expecting that just a few kilometers of a barrier between the Western Ghats, because elephants and even the other taxa that I talked about, except the high elevation species, many of them do come to much lower elevations. So why would they not just be freely walking across that? Again, perhaps it's a behavioral barrier, in the sense that it's a large tract of habitat that was very different from the habitats that these organisms occupied. It's been found to be a barrier for Nilgiri tar. No surprise, they're high altitude species. For those laughing thrushes and for Sholicola, no surprise. For high altitude, for middle high elevation frogs, no surprise. But it is a bit surprising, to be honest, that they've also been to some degree a barrier for elephants as well. So I think it's probably not true that they never walk across that barrier, but on the time scale of hundreds or thousands of years, that exchange of genes between populations is relatively low. We're at 1:05, and I want everybody to have a good lunch. So I'm going to ask to end this session. Vishu, do you want to come in and say anything so that you can start on time? Gokul, do you know if Vishu's here? He's here.
Yeah, I'm here. So yeah, thanks everyone for the very interactive participation, and so keep this on for the entire entire series of workshops. So if there are no more questions, Karthik, thank you very much, Karthik, for a wonderful session. And so join us back by 1:50. I know it's very short time, but we will begin only by 2. Start by 1:50. That way, admission takes a few seconds. Okay.
Yeah. Thanks, everyone. Thanks for participating, and thanks for all your questions.
Okay. Okay, great. Thanks. Thanks, everyone. Bye. Arun, please start the meeting by 1:45 and share the link. Arun, are you there? Okay.
Yeah, sure.
So you can stop the meeting now, and then just before that, make sure you start the next meeting by 1:45, and also send the YouTube link to Kanika, which she will then send it to everybody else, the YouTube participants. Okay.
Sure, sure.
Okay. Thank you. Bye.